how do I pronounce your name, Dr. Heike Wex? Is that correct?
Mercy, I think there is some echo coming in. Yeah. Hello, everyone. Today we are indeed having our next lecture for the cloud physics lecture series, and today's lecture is from Dr. Heike Wex. So I would like to introduce Mercy Varghese, from scientist from our institute. She will be introducing Dr. Heike Wex. Thank you.
Thank you, ma'am. Dr. Heike Wex research interests are focused on atmospheric aerosol and particularly include the experimental examination and theoretical description of aerosol cloud interactions such as hygroscopic growth up to high relative humidities, activation of particles to cloud droplets, heterogeneous ice nucleation processes, atmospheric aerosol particles as nuclei for cloud droplets and primary ice. Besides, she is concerned about climate change and the related issues, and she works for measures preventing a severe climate crisis on a more regional level in her spare time. Thank you, Doctor, for for agreeing to give us a talk. And I would like now Mahin sir to explain, give an abstract about Dr. Heike Wex's talk.
Mahin, you are not audible.
Yeah. Thank you, Marcy. And I welcome Dr. Heike for today's talk. And the title of today's talk is Ice Nucleating Particles in the Atmosphere. The abstract of this talk is as follows. In the atmosphere, in the temperature range between 0 and minus 38 degrees centigrade, the ice nucleation in the atmospheric cloud droplets has to be aided by a catalyst, which is provided by one kind of atmospheric aerosol particles, which is known as the INP, that is ice nucleating particles. Therefore, this INP is important for mixed phase clouds, but also for ice formation in the cirrus clouds. And the ice formation in turn is important for cloud radiative effect, precipitation formation, and cloud lifetime. INP comprise different types of particles, more specifically biological and mineral dust that is available in the atmosphere. And they are very rare in general, but still occur in vastly varying concentrations depending on factors on location on Earth, season, and temperature. Intensive laboratory research on INP was done in the past decades, while now the focus has shifted at understanding atmospheric INP based on atmospheric measurements. This presentation will provide some basic understanding of ice nucleation and measurement principles. We'll summarize the main findings from laboratory studies and then give an overview of the newest understanding gained from atmospheric measurements in the past years. Due to the vastness of the topic, it is thought as a spark that may kindle curiosity and further own research in this important topic. I welcome Dr. Heike to deliver her talk.
Okay, so I hope that all works well now, and I switched to my presentation. Is that seen now?
Yeah.
Okay, thank you. So very, very much thanks for first having me in the series, and then also for all those welcoming warm words in the beginning. And yeah, a warm welcome to everybody who is watching this now or maybe in the future. And as it was already said, I will talk about ice nucleating particles in the atmosphere today. While I prepared that talk, I realized that I aimed for too much. So I will try to stay in the 45 minutes range. Maybe I will be 5 minutes too long or something. I can really only touch things on the surface. But as already said, I will really try to give you an overview so that you can then dig into those directions further. You can have the slides later on. I even prepared a literature list that can be distributed via Tara, I guess, later on. So you don't have to scribble or make notes while you watch this. My overview also is kind of funny or strange. First, I will give very general introductions and then some basic knowledge. Of course, you don't know what that means, but you will see that later on. And the last part will be results from recent field studies. I show you that overview because these are the 3 big parts I will talk about, and they will all roughly be equal 1/3 of the talk. And I said all will need to stay on the surface. As we are in the lecture series on clouds and precipitation, I thought I spent 1 slide in the beginning on atmospheric aerosol particles, as I don't know if you all know about them. In the atmosphere, we have primary particles that are emitted directly as particles, and that happens during processes such as dust storms or sea spray production. But also when something burns, you emit ash and soot particles. However, when something burns, or from the biosphere, you emit also gases. And in case you wonder, this plot up here, this is an algal bloom from a top. So that is an ocean picture. And so those gases in the atmosphere can form particles just from the gas phase, or they can add material on already existing particles. So we have the secondary gaseous formed particles, and we have particle aging. Particles change while they are in the atmosphere. Particle concentrations are roughly around about 100 to 10,000 particles per cubic centimeter. Just very rough numbers, but you see this is quite high compared to typical INP numbers. They are very rare compared to all particles, with some 10 to 100 per liter, or even only 10 to 100 per cubic meter. You will see data on that later. So that was it about particles. Now about freezing. And first, homogeneous freezing. That is freezing without any particle. So we all know that below 0 degrees Celsius, ice is the thermodynamic stable phase. So water wants to be in that phase. But to form ice, an energy barrier needs to be overcome. And what happens, you can imagine with this, this circle here in the upper left side, that's supposed to be 1 droplet. It's 1 water droplet. And you have that little ice germ in it. This is a number of water molecules arranged in the shape of ice. Now, imagine that ice germ has that size here, like less than 1 nanometer in diameter. Now, if you add 1 more water molecule to that, you see what has to be done, or what happens on the energy side. You gain energy from adding that water molecule to the volume. That term here goes with r to the power of 3, but you need energy to form the surface. So that term here goes with the r to the power of 2. You have to account for both together. You get the blue curve. That's a calculation done for minus 30 degrees Celsius. And you see, if you have that germ that is smaller than that maximum here, if you want to add 1 more water molecule, it costs energy. It doesn't happen on its own. So, however, if that ice germ, that ice embryo, for some reasons gets large enough, like it was or is in here, that is the critical size. So the whole thing will start to freeze through, because from there on, by adding another water molecule, we will just always gain energy. So watch for that blue curve. That is now, that is in the next picture. It's that one here. That was at minus 30 degrees Celsius. That is the critical size at minus 30 degrees Celsius. That's the curve for minus 20 and minus 10. That is the critical size at these temperatures. You see, the critical size that original ice embryo has just gets larger the warmer it gets. And such big ice embryos just never occur randomly in nature. Only at round about minus 40, minus 38 degrees Celsius, this critical ice embryo is so small that a water droplet in nature will freeze on its own. So that is why we need an ice nucleating particle to make ice between 0 and minus 38 degrees Celsius. So in nature, you have surfaces everywhere that act as INP. But in the air, you really need these particles. And what these surfaces do is that water molecules arrange on them in an ice-like manner, and that helps to overcome that energy barrier. So we need a heterogeneous freezing. And so INPs act as a catalyst for that freezing, and there are different processes that are discussed for heterogeneous freezing. There can be deposition, ice nucleation, where water vapor from the air deposits as ice directly on an INP. Contact freezing means an INP hits a supercooled droplet, a droplet that's at below 0 degrees Celsius, and then it all freezes. Condensation freezing means below 0 degrees, you condense water on the INP and then it freezes. Immersion freezing is you condense water above 0 degrees Celsius, cool it further, and then it freezes. However, already in 2014, Claudia Macaulay started a discussion if deposition ice nucleation exists at all, or if this isn't rather condensation freezing, condensation of water in pores and subsequent freezing. So that's a possibility, and there is more publications on that from recent years. Contact freezing. A lot of nice work was done on that, for instance, by Alexei Kiselyev's group in for instance, that paper by Hoffman et al. 2013. But in the atmosphere, collisions are too seldom, so that probably doesn't play a big role. Condensation freezing. I had a paper on that in 2014, and I have much more data, just never managed to finish writing that up, that actually condensation freezing can be looked at as immersion freezing in concentrated solutions. You can use the same equation and just add a freezing point depression. And immersion freezing is the last that is still there now, and that has been acknowledged to be the most important freezing process in mixed-phase clouds in that temperature range between 0 and minus 38 degrees Celsius, where those mixed-phase clouds are. And there are some publications that said that already early on. So let's look at a cloud. Here we have a sketch of a cloud, and below you have the particles I talked about. And when an air parcel rises, temperature decreases, relative humidity increases. And at one point you have droplet activation. And here I want to explicitly mention that even mineral dust particles can be CCN. They can be activated to cloud droplets. Often they are considered to not be hydrophobic, but actually they are not. They always have a little bit of soluble material on them. They are quite large. They become immersed in droplets. So once we have our droplets, no matter now if they are formed below or above 0 degrees Celsius, upon further cooling, those droplets that contain an INP now can freeze. As said before, that's mainly immersion freezing in the mixed-phase cloud regime. Heterogeneous freezing can be important for cirrus too, but I'm not touching on that today at all. So there is now a multitude of other processes that happen in clouds. I'm very grateful for this lecture series, but I can't cover any of that. I will basically focus on these INP that cause this immersion freezing, and that in turn is important for precipitation formation. If we would go through that path, you would see that, and also for cloud radiative effects. But I said that's nothing I can talk about today in that framework. It's too much. Others probably will deal with that in that lecture series. So our topic for today is INP. Re-emergence of INP research. Fact is that INP were researched on a lot already in the past century. Like it started in the 40s, it was very intense in the 70s and 80s. There's a summary paper from 97, which summarizes lots of materials that can be INP in the atmosphere. But around about that time, for 1 or 2 decades, INP research quasi vanished. And it started again in 2010 with lab studies. There are some newer reviews on that. And there is a service that a person from our community, Naruki Hiranuma, provides. He sends around an email every month where he publishes new publications from a range of journals on the topic of atmospheric INP or INP in general. If you email him, I put his email here, I'm sure he includes you in this list. It's a nice pre-selection. And end of last year, he also sent around that table showing us that in the past years, since 2013, on average, more than 200 papers on that topic appeared per year. That explains why there would be so much to talk about, and I can really only present a small fraction. So that's the first part, summarizing it. I want you to remember that INP are needed for ice nucleation in mixed phase clouds. They are important in that respect for cloud radiative effects and precipitation formation. They are very rare among atmospheric aerosol particles. And immersion freezing is the most important process for ice nucleation in mixed phase clouds. Next part. basic knowledge about INP. That now tells you a bit about INP in general. And the first thing is mineral dust particles are known to provide ice nucleating particles in the atmosphere. And that picture here is from a lab study by Kiselyev et al. What they did, they took a slab of potassium feldspar, put that in an electron microscope where they could also enter a little bit of water vapor and cool the slab. And on the right-hand side, you see ice crystals growing on certain locations on that potassium feldspar. And on the left-hand side, those dots indicate where that freezing started, how soon after after freezing could have started. So you see the red dots, these are the most efficient ice nucleation sites. And it had been thought for a long time already that it's specific sites on mineral dusts that cause the freezing, but it's nice to see it in a picture like that. And in the paper, they even determined which lattice characteristics it is that cause the freezing and how that fits to the lattice of ice. So that's a nice study. Now you can imagine if it's certain sites that appear randomly on mineral dust surfaces, if you have a small mineral dust surface, a small particle, the probability of having such a site is smaller than if you have a larger particle. And you can see that here. This is the study that we did in our flow tube. You see from the yellowish to the reddish to the black dots, the particle size increases. And what is seen here is the frozen fraction, how many of all particles froze at which temperature. In that plot, it gets colder towards the right. Typically, it's the other way around. But anyway, you see larger particles have higher frozen fractions. That can be accounted for if you want to normalize that by looking at surface site densities, where you basically divide the frozen fraction by the surface area of mineral dust per particle. That was already proposed in a paper by Niemand et al. 2012. And for our data set here, you see the data of all the different sizes, how that nicely collapsed onto one line. By the way, I will not be able to talk much about parameterizations, or not at all, in this in this presentation. There's more to in the end in the backup slides. So if you get that ever, you could have a look there. So, but back to our mineral dust particles, that is one thing that was newly found in the last decade, and that is that among all minerals, potassium feldspar is the most ice-active mineral. You can see that here in that plot by Atkinson. I added more publications here. There are even many more out there. You see here the frozen fraction again, here that NS parameter. And for both the green dots, that is the potassium feldspar, are by far the most ice active. So that is the important mineral to look for in the atmosphere if INP are concerned. It's also known that biological particles can contribute INP that are very ice active. And for instance, here I show you bacteria, pollen, and fungal spores, but there are more like lichen and algae and probably mosses and many, many others. So the thing is, those original microorganisms have sizes of some micrometers, length and width. The pollen are the largest amongst them. So they are really big. But we know now, from research in the last decade, that on all of these, it is a small macromolecule of a nanometer in size, maybe roughly that order of magnitude, that causes the ice nucleation. So for bacteria, it is protein complexes in cell membranes. And for pollen and fungal spores, it can be polysaccharides, so long chain sugars, or also proteins. And for them, they can typically easily be washed off. So we know that from research in the last decade. I mentioned some papers here where with our flow tube in Leipzig, we got ice nucleation rates for single macromolecules, but I won't dwell onto that. I only want to show you one plot from Pune et al. What that shows is the size of that macromolecule versus temperature where it caused ice nucleation. And this is for some bacteria here, for some pollen and some fungal spores. And you see also that blue line. That blue line now is the calculated size that original ice embryo needs to have, like that critical size that I showed you in one of the first slides. And you see that the size of those macromolecules nicely fits to the size that the critical ice embryo needs. So how you can imagine that is you have that macromolecule on your huge microorganism sitting somewhere, and then water molecules arrange around that macromolecule, and from the outside then that looks like a huge clump of ice, like a huge ice embryo, and then the freezing of the whole water surrounding the microorganism can start. So that's nice, biological particles. How do we know if we have biological particles in a sample? So we all do know that it's not good for us to have a fever because our temperature rises and then proteins can denature. And that is what is used here to see if there are biogenic INP or proteinaceous INP in samples. That was, for instance, suggested by Hill et al. 2016. You see a soil dust sample here, how many INPs and INP concentration in the sample versus temperature where it freezes. Temperature here now, from now on, always goes down to the left, towards the left. Sample had a high ice activity at high temperatures, stayed like this if it was heated only to 60 degrees, but if it was heated to 105, all of these INP here were destroyed. Same was seen in samples from the UK, airborne samples. They had some samples that weren't very ice active, heating didn't do anything to them, but some were very ice active, and when they heated them, the ice activity vanished. It was seen in many, many more papers, but I still also want to show you that one from a Beijing dust storm, because just it's a nice paper just in general. And even for a dust storm, they found that at minus 10 degrees Celsius, 80% of particles across all size ranges, so these are different sizes from 1 to 10 micron here, were heat sensitive. And even at minus 20 degrees Celsius, still 30% of all particles were heat sensitive in Beijing, in dust storms in Beijing. So we have many biological INP out there, active at high temperatures. Now, how do we measure that? There are two basic kinds of methods, in situ and offline. For in situ, we have a different zoo of instruments. So this is the flow tube we are working with. It can't be transported, and it is a very special way of working with this. But then there are also things that can be transported, like CFDCs, or now a very new and expansion chamber called Pine. They all have different operating principles. I originally included them here and threw them out, no chance to discuss that, but you can read the publications that I put here to see how this all works. They typically have single particle optical detection, and they have some advantages with that. They have high time resolution, and they are closer to what happens in the atmosphere, because typically they insert the aerosol and some water vapor and cool it. So you have the same processes going on in the atmosphere. They have disadvantages too, because they have high detection limit. A single ice crystal or particle doesn't scatter much light. So it's really difficult to measure enough. And also the amount of air you drag through there is limited. And they are more difficult to operate. And for large particles, because they usually discriminate or detect ice by the size of what comes out of them, they already cut off large particles at the entrance. And that could be a disadvantage in some cases. Methods, offline methods, they have like it feels like 10 years ago no one had them, and now like there are hundreds of groups who build some of their own offline techniques based on a few principles. So we built these two that you see here, also based on literature data that we found, and also thanks to support from the colleagues who built the first versions. And what we have is one where we put 1 microliter droplets on a small glass plate, 90 of them. You see that from top here, and cool that by a Peltier element. And then we have another technique where we put 50 microliter droplets in PCR trays. We all know PCR trays now. When we started 2015, I had never heard of them before. But anyway, so we all put 50 microliters in each of these tubes, and then put that in a thermostat and cool it. And in both cases, we take pictures from top once per second. So what you can examine in them are suspensions. So you can have water and just put a spoonful of soil, for instance, in that and make your droplets from that. You can collect rainwater or ocean water directly, put the droplets in or on there, measure. Or you can collect airborne particles on a filter, wash them off into water, and then make your droplets. And with that, you can get atmospheric ice nucleation particle concentrations. You can even punch out filter material and put them in the PCR tray together with the water. That's what Pune suggested. So, however, you need to work very cleanly, determine your backgrounds and everything. So there, it sounds like easy. It's not. You really have to test a lot and get your background down. And that is because you always have a little bit of contaminations in the water. The larger the droplets are that you look at, the higher are the INP the lower are the INP concentrations that you can measure. You get the more rare stuff, but also the more contamination issues you will have. So all in all, you have the advantage that you can measure low INP concentrations. You can also do additional analysis with the samples you have there. Even still in the tray, once they were frozen, you can still do stuff with it. But you have that contamination issue, and you can go down to smaller and smaller particles, but that causes other issues. And you have long sampling times. That's an advantage. You can get the more rare stuff, but you have a bad time resolution if you do that. So advantages and disadvantages. And now imagine you have your sample with the droplets, you cool it down, and what you get is a curve like that one. While you cool it down, more and more and more droplets will freeze, and you typically take pictures and count them. And so the frozen fraction just increases as temperature decreases, and you get a cumulative distribution. Now in a droplet that froze very early, there could be other INP that would have frozen later, but you will never see them. And that is why you have to assume that your INP follow a Poisson distribution over your sample. And that was already understood by Wally in 71. And if you use that Poisson distribution, then that is the formula that you use to calculate the average number of ice-active entities per droplet. So one example, if you measure like 40% of all of your droplets froze, that formula tells you that on average you have 51 of that. At that temperature, the average number of ice-active entities per droplet is 51 on average. So that is what the Poisson distribution accounts for. And now once you have that, you can normalize it to different parameters. So for instance, if you know how much how much air volume you sampled on a filter, with how much water you washed it off and how much of that water you put, what's the volume of one of your droplets, you can calculate the volume of air that was sampled into each droplet, so to speak. And if you use that formula, then you get the INP concentration per volume of air. Or if you just collect rainwater, that would just be the volume of your droplet, and you would get the concentration per liter of rainwater. So that's how that is done. Now that we measure, we know we have different instruments. Where to measure with which instrument? And here in that plot, again, we see temperature here on the x-axis and atmospheric INP concentrations that we now know how to obtain. That blue-white striped area gives us roughly observed INP concentrations, just an idea on where they are. And that striped area appears to range where CFDCs measure. They can't measure at the very highest temperatures, and they have that lower detection limit I talked about. The orange is for cold stages. They can measure very well at high temperatures down to low concentrations, but the colder it gets, the more issues they get with contamination. That's why the orange fades out here somewhere. So that plot actually was given in situ et al. But it's just generally valid. And that now shows you an assembly of measured concentrations of INP from precipitation, published 2015 by Peters and Wright. They collected data from other publications, measured some of their own, and got all of these different freezing spectra. So one line here would be one freezing spectra for one measurement. And that gray area here, that's enveloping their data, you will see that again and again in later plots now. And interesting thing here is, from all that I told you before and what people understood very clearly in the last decade, we can now say that at the lower temperatures and higher concentrations, it's the mineral dust particles that are ice active, that show up there because they are more numerous, and they come from deserts or from soils. But that bump that we see up here, that is caused by biogenic macromolecules. And so that is the general overview. And so with that, I come to an end of that summary section, on a bit more INP information. And I want you to remember from that in the atmosphere, mainly two main INP types contribute, which is mineral dust particles and microorganisms. Microorganisms have macromolecules which cause the ice activity, and they can be proteins or polysaccharides. And they are very ice active. You saw that, it's that biobump at the high temperatures, but they are very rare. And heat can destroy the proteins, so that is used to see if they were present in the samples. Mineral dust particles have ice active sites, and they are ice active at lower temperatures, but more abundant in the atmosphere. However, more abundant means they are still rare. And potassium feldspar is the most ice active mineral dust. And I told you that a multitude of instruments exist to measure INP either in situ or offline, and they have all their strengths and weaknesses. So should you ever consider starting to measure that, you should really think about what you want to find out, because that will determine what you want to use as an instrument. And as I already said earlier, the topic of parameterizations of INP data has to be left out here. So I'm running a bit late, I'm talking too much. Anyway, so now I come to the third part of my talk. Upcoming now are results from recent field studies. And the thing that we are all looking for who do that research these days is that we want to know in which locations we have which INP concentrations, and where do these INP come from? So basically sources and occurrences. In the following, I will throw 31 publications at you from the time from roughly 2013 until 2022. Actually, when I had finished preparing this, I counted that and made that little figure here, because I realized that most of the stuff I will show you now is really very, very new. Most of it comes from the last 5 to 6 years. I will really give you only a few main messages. And just as a summary, most of the results you will see in the next slides come from offline methods, and it's basically all still ground-based measurements. So we do not have a very good understanding yet of what happens higher up. There are some publications. There is a lot of research going on in that direction right now, but that is for the talk in 5 years to present, I guess. So what do I want to tell you? One thing is that highly ice active atmospheric INPs are often supermicron in size. The first publication on that I know about comes from Mason et al. In 2016. You see that over here. So they give the fraction of supermicron INPs for different locations at different temperatures. The locations were in the Arctic, in France, in Canada, and in the US. And at minus 15 degrees Celsius, you see that across all sites, roughly maybe 80 or more % of INP are supermicron. That now fits, because I told you it's at these high temperatures, you have the biological INP. They are large microorganisms with tiny macromolecules, but still, that fits. Even still at minus 25 degrees Celsius, a high fraction at all sites was supermicron. And here now you see that this might be a problem for the in situ measurement devices. However, they often aim at measuring at the colder temperatures where the concentrations are higher anyway. So at that location, at colder temperatures, I don't know these fractions. And then Cremian et al., you see a picture of her work later on. She also found that larger particles are more ice active. And it was also found at Cabo Verde, Cape Verde Islands, close to the Sahara. They are also across all temperatures, roughly 80% of all INP were supermicron in size. Next topic. INP and anthropogenic pollution. Had I talked to you 10 years ago, probably I would have said, yeah, it is assumed that anthropogenic pollution contributes INP, but that is not the case. So we cooperated with Beijing University. They collected filters, and then Jay Chen came over and measured them, and we published that work together. And you see on the right-hand side in the background, the Patterson Wright background, as I promised you, you'll see that often. And then the green and brown are data from our 2 cold stages, the one with the larger droplets measuring at the higher temperature. And during the experiment, the conditions were varying wildly from clean to very polluted, but you see all these lines are very close to each other. And if you make a cut at minus 16 degrees Celsius, you see those concentrations are shown here versus different aerosol parameters, black carbon, PM2.5 mass, total number concentration. There's just no correlation. And so we said that Beijing air pollution did not contribute INP down to minus 25 degrees Celsius. However, just one note, these concentrations shown here aren't always valid for Beijing. I mentioned before another paper by Chen et al., that's a different Chen now here, where they saw that they can have dust storms in Beijing and then the numbers increase. But we didn't have a dust storm during that air pollution study. It's just a note of caution here. So the other thing is Tarn et al., they measured during a night with bonfires in the UK, and you see concentrations in the afternoon before the fires, concentrations in the night of the fires, they were similar. So the same conclusion, no contribution from the fires. Others concluded that as well. Belty et al. Says there was no contribution to INP from ship exhaust, maybe below minus 36 degrees Celsius, but that's almost outside of the mixed phase cloud regime. Yadav found no contribution of INP from local pollution in northern India. That's from precipitation samples. Tobo et al. Had a year-round data set in Tokyo and also found no anthropogenic contributions. They said variations they found came from long-range dust transport or from biological INP. But I also want to show you this study. That is from 2017, and they said they saw an enhancement of INP due to ship emissions in the harbor of Gothenburg in Sweden. However, my argument here would be what they find is really, it's at minus 30 degrees Celsius, they find an increase of 2 INP per liter. And if you would compare that to the typical Patterson-Kreitenweiss curve, you will see that more often in that presentation. You will see that's way below where the typical concentrations are. So even if there would be that contribution, it would not be noticeable. So maybe this explains that kind of contradiction. So in general, I would say anthropogenic pollution does not contribute INP. Next topic, and I take a sip of tea. So, INP in the Arctic. Here now you see the plot by Cremian et al. That I promised earlier. This is again, as always, INP spectra. Those are for the smallest particles she looked at. She did it size segregated. This is a bit larger. Here we are now in the supermicron size range, and here it's even from 3 to 12 microns. And first you see that the concentrations increase with size, as I said before. You also see those colored curves. They come from later in their experiments. So they were in the Arctic from March until May. And towards the end, they found these high concentrations for the large supermicron particles. And we actually found the same in a study where we looked at filters from 4 Arctic stations. And for all of these stations, we have plots like that one here. It's again INP spectra. We found low concentrations in the winter month. It's January through March, and high ones in the summertime. It's a bit different when this high concentration started. Here it already started in April and was seen until August. And we also tried to find out where these highly ice-active INP could come from. And this is now 3 maps of parts of the Arctic for different cases. And you see here the shoreline of Greenland, a bit of Svalbard, a little bit here in the ocean. Again, a lot of Greenland, northern Greenland, bit of Canada. This seems to be terrestrial, and this is a lot around Svalbard. The location is always where the samples were taken, this this little black indicator here. But honestly, we couldn't say if it's terrestrial or marine sources. And you will see towards the end, no one can tell you, still not. That's a big puzzle, an open question. So we have these high concentrations in the summertime. So now we thought, let's see if it changed over the centuries. And there are 2 studies that looked at ice cores. So that is by us, and that's by a group in Frankfurt. They went some 100 years back, different for both studies, but both only went up to 1990. And they both said that they did not find an increase of INP, annual INP concentrations, no annually averaged, or even more than annually averaged, in that timeline. And that now isn't, and only I should mention, I'll say that maybe there's a small increase after 1960 due to land use change. But we do know that there's Arctic haze in the Arctic, that is anthropogenic pollution, summing up more and more and more during the winter. So right now, the concentrations of Arctic haze, anthropogenic pollution, should be at their highest. And when then everything starts to melt, precipitation sets in and washes that out. So that kind of indicates that, like, we have these high summertime concentrations. That is against the circle of Arctic haze. We have no increase over the centuries. So again, one indication that anthropogenic pollution did not contribute INP in the Arctic. However, a note of caution, these studies with the ice cores only went up to 1990. And there is Arctic amplification. The Arctic warms much more than the rest of the globe. They are up to 3 degrees Celsius, up to 5 or something already in some locations. It's really dramatic up there. And that means the whole biosphere could change. But the Arctic amplification only started to really become visible around about 1990. So that the signal of that wouldn't have been included in these ice cores. So there are probably many changes to come in the Arctic also related to INP, which also then changes clouds. Where could be sources? There are possible terrestrial sources. I give you a number of papers here that talked about possible terrestrial sources. So here you see, for instance, there are dust plumes coming off Iceland. Here are possible dust contributions either only from Svalbard or from all land sources in the Arctic, from these lower papers, from these 2 papers down here. And all in all, what these papers say is that there are Arctic dust sources that can contribute. They are connected to biogenic INP often, so there might be material attached on them, or maybe only the macromolecules washed off from other microorganisms, then going on to the dust. Can happen. Biogenic INP, like bacteria or pollen spores, can be transported in the air directly, or it can be long-range transport, as said in this paper here by Seardai. So there are many possible sources for terrestrial INP in the Arctic. There are also sources for marine INP in the Arctic. Irish et al. Found that in the Arctic Ocean, INP just swim around there, particularly many close to the ice edge, where there's melting sea ice. Hartmann et al., one of our studies, we flew over the ice at that time of the year in 2018. And when we flew higher up, we didn't see enough INP to really detect them. But when we flew very low, we found these colored curves down here. Those were the 3 flights, like 100 meters above the ice only, with high concentrations of INP. So we suspect that these came from open polynyas, that they are a source, however, only very locally detectable. So it's a kind of a weak local source. And now with that paper, it's really new. It's still in press, basically, but it kind of, I don't know, I can't really fully comprehend what's going on there, because that's by Porter et al. They went to the North Pole, to the region of the North Pole with an icebreaker, the Odin, and measured INP. You see it on the right-hand side, again, petals and right in the background. All the colorful plots are from a lot of different studies. Their own data in that plot are indicated as black stars. And you see at the North Pole, the highest concentrations trail up that curve from Peters and Peters and Wright. And it's like, whoa, these are high concentrations. And they connected those highly ice active samples with back trajectories. Those are the yellow ones here that come from that Kara-Z region. So they claim they have a strong marine biogenic source that contributes those INP. And yeah, say, why not? And now I'm introducing a puzzle to you that we need to solve, and you will stay puzzled after I'm finished. So that seems to be a very high source, but one possible reason could be you have rivers running into that region, and we do know that INP can be washed off from soils into rivers and then transported into the ocean. That's true, but still, the puzzle is that we have two studies, one at the Cape Verde Islands, the other one in the Arctic, where we looked at INP in seawater and in the air at the same time. And that plot shows what we measured in the atmosphere. That's the data up here. And the colorful red and blue dots down here are INP concentrations expected from bubble bursting, so from this oceanic particle generation mechanism, based on INP concentrations we measured in the seawater. So this is the case for the Arctic. The same picture was found for Cape Verde. And there are orders of magnitude between these two data sets. So unless there's an enrichment of marine INP during the bubble bursting, like many, many more get out than, for instance, sea salt gets out. The marine sources here, at least in these two studies, cannot explain the airborne concentrations. And now you see why I'm so puzzled by the Porter paper. Now we go to a place where that information really fits, and that is remote oceanic regions or the Southern Ocean. McCluskey published 2 papers in 2018, where she found very low INP concentrations in the Southern Ocean and in the clean Northeast Atlantic. So that is now not, the gray here is not Peters and Wright, but it's similar to that. Low concentrations. Now, that fits to the fact that, well, marine processes maybe only contribute little amounts of INP. But again, I can't see where the Porter paper fits into there. And I'm not saying it's right or wrong. I just say it's amazing. And Southern Ocean seems to be clear. There are low concentrations. Others found that as well. For instance, Welty, they collected some data their own and data from other publications, from ship measurements, and when they had continental air masses, INP concentrations over different regions across the globe were higher than the marine INP concentrations, and particularly lower concentrations in the Southern Ocean. That seems to be safe, and that was also reported by 2 other papers that did work around Antarctica. So that's clear. Not many INP in the Southern Ocean, and that fits to an observation that has been often described based on satellite data. That is that in the Southern Ocean, we often have high concentrations of supercooled liquid droplets because it's assumed the Southern Hemisphere doesn't have so many terrestrial sources for INP, be it dust or biogenic INP. So droplets just don't freeze in these clouds there. So that part now gives a nice picture where everything seems to fit together. And now I come to the end. I'm already a bit too long, but please give me time for 5 more slides, because I also want to give you some impressions from newer long-term studies that are now coming out. And for instance, there is this one, Schneider et al. From a boreal forest in Finland. They measured for over a year. Now, one INP spectrum here would be one of these lines. You see the concentration again here as usual, but you see the temperature where freezing happened color-coded. Actually, it's a pretty clever way to show that. And so you do see there is a cycle. It's like higher concentrations in summer and lower in winter and going up again in summer. They compared their concentrations, INP concentrations with a lot of different parameters and found no correlation. And so they suggest one could parameterize INP with respect to temperature. And in a way, that's the same that was also reported for the Arctic, like these high concentrations in summer, low in winter, and that we still don't know the sources or cannot really get a good handle on them. And now there are more long-term studies, for instance, from 4 different stations in the Arctic, Central Europe, the Caribbean, and the Amazon. For like almost 1.5 years, some gaps in between for some stations, but nevertheless, this study says they did not see long-term trends or seasonality, and that the short-term variability was much larger. However, they only measured at -20 degrees Celsius and -25 degrees Celsius. So that is that temperature range where the biogenic particles may not contribute so strongly, or not in large numbers anymore, or not detectably large numbers. So that might explain that missing seasonality that they see, because for the Arctic, for instance, we saw that clearly as one. They also see things that by now should seem familiar to you. Amazon was mostly unaffected by biomass burning. There was no anthropogenic influence in Europe or the Arctic. And the Caribbean was affected by long-range dust transport. So that all fits to what I think we understood in the past years. And then more long-term studies, one by Testa et al. They measured for 7 months in North Central Argentina. You see the data here. Again, the Patterson-Wright plot. Now it's yellow in the background. Their data as black dots. Another background here from the Kanji review paper. And their data trail the Patterson-Wright again very nicely. But they also do not see a seasonal cycle that can be seen here. And that is now even true at the high temperatures. They saw that those very highest active INP were heat liable. They were biogenic. They also saw for the lower temperature ice active INP, they could be destroyed to a large fraction by H2O2 treatment, which means they are probably organic and not dust particles. There were some dust particles that corresponded in concentration to those organic INP, but they were very, very, very small amounts only. So they say probably the INP they observed were regional and from arable topsoil. They also found one thing, that is the biogenic INP, those up here, they peaked during rain and high relative humidity. So it could be that the surfaces, terrestrial surfaces, emit INP when rain falls on them. And then one last study, Gong et al. That's now a study that we did that was just published on ACPD a few days ago. That is from southern Chile, where we measured for 11 months, 8 kilometers west of Punta Arenas on 800-meter high mountain. And here, a bit like in Argentina, we found surprisingly high INP concentrations. And northern Argentina, I wouldn't have been too surprised, but there we are really at the tip of Southern America. I expected to see the clean Southern Ocean conditions, but no. You have like 200, 150, 200 kilometers of like island land before you reach that site. But the concentrations here are, again, as they are in Patterson-Wright. And the samples here are color-coded with respect to season, but don't look too hard because we also did not see a seasonal cycle. And so that was amazing. And a bit like in Testa also, and in other studies, we found that across all temperatures, there was a very high fraction of biogenic INP, again, like 80% or higher, down to -16 degrees Celsius. Below that, only half of the filters contributed to the data, so we didn't show the numbers anymore. But still, very high concentrations are biogenic. And we also found that precipitation seems to be the only thing that can explain why sometimes INP are higher. And then I remembered that I read that a long time ago in a paper by Huffman et al., who found that already for a forest area in the US. Also, INP and bacterial or microorganism enhancement in the atmosphere in general during rain and high relative humidities. That could hint to a source or to times when emissions take place. So, summary for results from recent field studies. I told you in the beginning that a large number of atmospheric INP are supermicron in size, down to -25 degrees Celsius. No INP from anthropogenic pollution is typically observed for temperatures valid for mixed phase clouds. Annual cycles of INP were found in some locations, like the Arctic or the Finnish boreal forest, but not in northern central Argentina or southern Chile, and also not for lower temperatures in that one study I showed you with the 4 stations from Schrodt et al. Summertime Arctic can have INP concentrations as observed over mid-latitude continents. Remote marine regions clearly have low INP concentrations. Also, the clean Northwest Atlantic showed that. INP from sea spray production cannot explain atmospheric INP unless there is a real strong enrichment during the particle generation process. It's the bubble bursting process, if you want to look that up. And there are enhanced INP concentrations over continents, and maybe that happens due to precipitation events. That, I thank you very, very much for listening to me for so long. I hope you found it interesting, and I hope it sparkles your interest in the topic, because there's still large amounts to find out. Thanks.
Thank you. Thank you so much, Heike, for this exciting talk. And maybe, Mahin, please see the questions.
Yeah, we have many questions. First,
okay, let me change my system here a bit.
Let us take a few.
Yeah, now
breaks.
I hear you. Now I hear you good. Should I stop my presentation?
Yeah, no problem.
Yeah, I stop my presentation. Yeah.
Yeah, I hope nobody wants to go back to that right now. Or
probably could do that, yeah. But now I at least see you.
No problem. You can share it. So someone wants to refer to something.
Exactly.
There are plenty of questions. For
discussion. They will start from Dr. Anupam Hazara's questions. He's asking, can we do immersion freezing nucleation in spin? You know, spin is a DMT instrument, that is spectrometer technique. And normally we inject dry aerosol particles in spin for studying deposition nucleation.
So let me repeat. The question was if immersion freezing can be done in spin, because the dry aerosol is injected. Yeah.
Yeah.
So the way how spin works, or all CFDCs, if you have these 2 walls that are coated with ice, and they are at different sub-zero temperatures. One is a bit colder than the other. So you have water vapor diffusion from one wall to the other. And in the middle, a maximum supersaturation forms that depends on the delta T between the 2 walls. So you can increase the delta T between the 2 walls, and with that you can increase the maximum supersaturations that the particles will see. So that means that yes, you can cause conditions where droplet formation happens. And then that's a bit of a question of definition, because in some years ago, like 10 years ago, people said, oh, we call that condensation nucleation because the droplet formation happens below 0 degrees Celsius. My argument would be, when your delta T between the 2 walls is big enough that you have a supersaturation with respect to water, and that depends also on the absolute temperature, but that can be calculated, then you have immersion freezing. So I would call that immersion freezing. You will see it in some papers that it's very carefully then called condensation / immersion freezing. But I would argue, yes, you can do it. And the water vapor comes from the ice from the walls. It's in the instrument.
So his second question is, if we collect sample from different locations, how can we test those samples in spin?
Oh, you mean you want to collect samples like on a filter and then re-immerse them in spin?
Yeah.
That is something that I find quite difficult because I showed laces, but I didn't talk much about it. And the thing is, at one point we understood that if you do that, like you collect outside samples and regenerate them, the concentrations are so low that it's very, very difficult to see anything in these online instruments. Because they drag through a number of liters per minute, but even if you generate particles, I mean, you will not have so many INP in these generated particles, and you lose them. The thing is also they are big, maybe. So in your setup, it will be very, very hard to introduce them in a way that you don't lose them in any bends. So my lab experience tells me if I have particles larger than 3 microns, and I go around a bend, they are gone. They all impact in a 90-degree bend. I should also say like the diameter of the tube and the flow, but still, I mean, whatever. Supermicron particles are hard to generate anyway. And yeah, we never tried it because we thought it wouldn't work. So sorry.
Yeah. Maybe, Heike, I want to say something here, that we have done something like a back sampler for the aircraft observations. So there is a possibility to do that with a back sampler. But it was very specially designed for the spin and for our aircraft campaign. So we have got some observations that way, but it is not a lot and a lot of effort behind it, because there is a time delay and everything matters into. So there was a real challenge to get it done. And yeah, and actually relating to Anupam's question regarding the immersion freezing also, with spin, it is not really possible to go to warmer temperatures, right? So there is a limit, right? Actually, we are missing that region which is most important for our clouds, warmer temperatures, like minus 5 to minus 10, minus 15, like that.
Yeah. That is, remember that plot that had that lot of orange in the background and that white blue striped area? That is where I stopped at minus 10. And this is technical reasons. These instruments just can't go higher, because also then you would need so high delta T that it just doesn't work anymore.
The range that you have shown is quite large there. So the variability is quite large to see. So maybe, yeah, after the questions from the audience, maybe we will have some discussions in there are several questions for you.
Yeah, and I should also say that we do have a spin. We used it for a while, but I never used it myself. I only was there using laces when we had visitors using CFDCs in general. So I think, Tara, you are more the expert on spin than I am. So maybe I will give questions to you on that.
And see, imagination takes us to different innovations. So we are really learning. So I just wanted to mention that there is a
it's totally great. Thanks.
Please go ahead with the other questions.
Yeah, there are 2 more questions from Anupam. Another question is, biogenic aerosol act as INP in warmer temperature. Do you think bioaerosols may play a significant role on the formation of cloud and precipitation when storm height is not very high?
Yeah, so that is a very short answer, yes. Also, very high isn't really defined here, but especially when the height is not very high. And I'm not familiar with conditions in India, I have to admit, but I do know there is a paper, and I had wished to include it, but didn't find a way to do it, by Gary Meller et al. Not Gary Meller. Grieche. Gary Meller is the one who describes spin. No, but I mean Grieche et al. That came out last year, I think. You can email me, or I can include it in my reference list. And what they did, they measured in the Arctic, and they found that clouds were different depending there's some noise here, I'm a bit disturbed, but anyway, if there is coupling to the surface or not. And when there was coupling to the surface, that means like there was a possibility to transport INP from the surface to the cloud heights. These clouds diluted sooner. They weren't as stable. They formed ice, and they didn't have so long lifetimes because the ice forms, then the ice grows on the expanse of the water droplets and falls out. And there was a clear difference between surface coupling or not. But those were in storm clouds. In the Arctic, you have these stratiform clouds. But if it works for those, I'm sure it. I still think for storm clouds they will influence.
Yeah. So there's another question from him. Whether INP concentration enhance or suppress precipitation? Is there any tipping point?
Oh, that is a good question. You may have realized that I talked a lot about aerosols, so actually not really clouds. And I have to admit that this is something that I can't answer with a lot of confidence. Because what you do, I would even assume that depending on the cloud conditions, you could have different answers. But really, I'm now on very thin ice. I can't give a very concrete answer. It just also depends like on the whole mechanism that follows up. So if you would have a relatively warm cloud and you form first ice, that first ice is important. But then you get the secondary ice processes, and they might depend on temperature as well. So, and that is, I think it's not so easy to give that answer, and I would even dare to say that no one can say that these days. Some models might have outputs, but I'm not sure if anyone can answer it for sure. Sorry.
Yeah, there are two more questions from Max Aragon. What is asking, is it possible to measure or estimate INPs from space or say satellite? And why INPs are very ice active? Does cosmic dust contribute to the INP population?
So the first from space, there has been a study that was introduced in an INP colloquium. I don't know if you know this INP colloquium that the ETH organizes once a month. And that was the second last or third last talk by should know his name. Well, I'm just not. Anyway, I can also include information on that later. And they looked at satellite data, and but not directly. It would be an indirect thing on how do clouds behave. So you can't really measure them directly. They are too few. You won't see that. And the second half of the question I didn't understand quite well. Can you repeat it?
Yeah, why bio-INPs are very active, ice active? Why bio-INPs are very ice active? Does cosmic dust contribute to the INP population?
Yeah, well, that is something that I think we will hear more about as we hear more about results from aircraft campaigns. And the theory was there in the past. And the thing is, even if cosmic dust would contribute, it would contribute in that colder temperature range, because those macromolecules that are made by the microorganisms, they are very, very special. And they are already, as I showed in that one plot, they are already very large. They are already providing almost the size of the critical ice nucleus. Dust that trickles in from above will probably be much, much smaller. And yeah, but we will hear more about it, as I said, when we have more aircraft campaign results in the future. So maybe some of my non-answered questions here can help push people into certain research areas.
Yeah, he has another question. If precipitation enhance INP concentrations and INPs promote precipitation, is it considered as a feedback loop?
That would be a feedback loop, yes. Yeah. Mm-hmm. However, I said earlier, I can't really tell you if it does or not. So, but that would be the description of a feedback loop, yeah. Mm-hmm.
Thank you. Yeah, there are no more questions from the audiences.
Maybe I can say one more thing that I have, like one thought that came that I have in my mind, and as I'm not a model, I can't show that. But what I think in the Arctic you have clouds 80% of all time. It's a very cloudy region. You have these stratiform clouds that seem to live forever. They just don't vanish. It's not quite clear why they are so stable yet. However, one idea could be that there aren't so many INP up there. Now, if you have a prolonged growth season because the ice vanishes and you get more INP in summer, you could make those ice vanish in the summertime. And throughout the years, the clouds warm the Arctic, but especially in summertime when you have some high sun, then the clouds actually have a cooling effect, only for a very short time. But that is the time when the biogenic ice, when the biogenic INP could come into play and dilute the clouds or make the clouds vanish. So you could have an additional heating effect there. That is just an hypothesis I have in my mind, and yeah. So if anyone wants to work on that, I'm not following it up directly these days, but I think others are doing it.
Yeah, Heike, you really touched upon many aspects of many observations and many aspects from all around the world. Really very encouraging for many, even students, those who are listening, to take up some of the topics that you mentioned. And I just wanted to ask you regarding this, the range of INP number concentrations that range from 10 to the power minus 4 to say 10 in the warm temperatures, and which is a very, very big variation. And is it how often it is indeed related to the after the precipitation, or because you have very clearly stated that after precipitation there is an enhancement in the INP also. So how often these observations are indeed related to such effects, which are, because all these are indeed counting for many interactions are coming into picture. So just wanted to ask that, as well as one more question I have is regarding the black carbon, aged black carbon. There are some studies showing that aged black carbon are also playing a role as ice nuclei. I think, yeah, that.
Okay, so the first was the correlation with INP and rain. So I guess that the sources aren't totally clear, and particularly the emission of INP from, be it terrestrial or marine sources, are both still not totally clear. For marine, we know that we have this bubble bursting process, like organic material or collects at the surface or enriches at the surface and enriches at the area around bubbles. And when a bubble rises in the ocean and bursts, you make particles. So that is one mechanism. However, I told you we need an enrichment to explain atmospheric concentrations. On the in terrestrial cases, you can of course have dust storms, like wind can pick up particles, but it's not totally clear. For instance, these large bacteria, why would they become airborne? Will they just be transported by wind? And these emissions from the biosphere into the atmosphere, that is something biologists know about. I just saw a talk 2 weeks ago where a person talked about these emissions, where someone modeled that worldwide. So that is some area where I'm just starting to learn things. And this correlation with precipitation, it has been found. But I also think I read somewhere, but I don't recall where, that if it had rained, there's an increase in the beginning, but if it rains more, it doesn't increase anymore. Maybe the sources are depleted. So that is still a very, very open field as well. And yeah, as I said, we want to learn about those sources and things, and we aren't there yet. And about those other studies, I just summarized what I know. And I said there are so many papers that I don't really follow everything. So maybe you can talk about that a bit yourself, like those where you see the soot contributing to black carbon.
There were, I remember seeing some of the papers earlier. I think Mercy has some question.
Yeah, Mercy has a question.
Yeah. My question is somewhat related to what Dr. Tara asked. We have seen that more like anthropogenic pollution is going
Oh, I think she has some connectivity
droplets.
Nancy, we didn't hear you. Maybe you repeat your question.
Okay, sorry. What I was asking is a follow-up of, you can say, like a sort of a follow-up to what Dr. Tara asked about black carbon. My question is, like, we see anthropogenic pollution, giving rise to smaller droplets, deeper clouds. Then you have this mixed phase clouds and all. So there you have the supercooled droplets. Then if we see, or if the pollution is not going to give us is not the INP, what will be the starting point of having this ice phase emission, right?
Last part, please repeat. Last part of your question.
You get chopped out. I don't know. Maybe you can turn off your picture, and then the transmission is better.
Okay. So like in when there is pollution, we tend to get smaller droplets, deeper clouds. Then we get the supercooled droplets. Pollution is not the INP. What is the starting point which creates ice in these mixed phase clouds?
Aha. Okay. Yeah. Just to repeat that I get it right, you say that, like, you do know that black carbon produces more droplets which change the cloud, but if this isn't the starting point for ice, what is? But you always will have some INP in the air, even in very polluted air masses. Although they are still a very, very low fraction, you will still have some there. There is this interesting observation that below roughly minus 20 degrees Celsius, number concentrations of INP and ice crystals in clouds agree, while at warmer temperatures, the ice concentration in clouds stay at the same high level, whereas the INP concentrations are much, much smaller. So you have all these secondary ice processes, which is a whole research area in its own. And you only really seem to need a few INP to kindle that ice that secondary ice nucleation. And so, yeah, I would say there are always some INP that can do that. But if Tara knows more about those studies on black carbon, maybe you can tell us a bit about it.
Yeah, I will email you. A long time ago I have read that, and yeah, I'll email you on that. I think Mahin has some more questions.
Yeah, just one question I will ask. I just came from a place. You have dust aerosols, and it had a lot of biogenic aerosols, and then a lot of smoke, a lot of pollution. I mean, you name it, all pollutions are there. And it is also a place where a lot of lightning strikes are taking place. So how we can measure those INP and separate those biogenic than dust or whatever? I mean, different kind of aerosols, pollen grains and all. Is there a methodology? And also, is it depend on the sample volume that we collect during an experiment?
I mean, if I had a location where I know I have a lot of dust and a lot of biogenic particles, I would still go for filter sampling and try to make that comparably short. And then you would have to see which kind of cold stage you are using, how long sampling times you would need. And what we typically do is we always measure our PCR trace, and then we heat them and measure them again. So with heat, you can already learn how much biogenic INP are there. There is one paper, Worthy et al., and they suggest that adding ammonium sulfate to your suspensions will increase the INP activity of dust. That's something we are just testing. And I'm not aware if others have tested that as well. And then there is an H2O2 treatment, hydrogen peroxide. And that was often done by groups, when you Google Hill et al., he's always doing that. And we wanted to start doing that as well, but haven't done it. But that tells you if there are organic materials in there. So you can find out how many proteins and what is the concentration of proteinaceous INP, what is the concentration of organic INP, and if that functions with the ammonium sulfate, you can find out how much mineral dust contribution you have, and that would add up most of it. And now about the black carbon, I said they don't contribute. So if they do, I need new answers. But so I'm really looking forward to reading this other material from you, Tara, that I'm not familiar with.
Thank you.
There was a paper by Sindhya Tuhi in which they found a positive relationship between this black carbon biomass aerosols and the ice concentrations. I can give, I think, I give that link.
Yeah, I do know about studies where biomass burning was examined, and it was said biomass burning might enhance INP. I also know of studies where it is known that biomass burning picks up additional dust particles from the ground, because you have that effect, the air is warmed, it goes up, and it drags with it particles from, like dust, soil dust particles from the surrounding area. So it could be, is one thing that I have in my mind as answer to that, that you just enhance dust concentrations in the air, and with that you increase number concentrations of INP. But then you would also really have to see if those are biogenic or not. And you can, of course, always do chemical analysis to your samples. If you have like filters and wash that off and have enough material, you can always do chemical analysis. That's the nice thing. We've done that a couple of times and never found any correlation, but we haven't measured in such a wildfire environment yet. So there you could just run typical chemical analysis to see if there are correlations or not.
Thank you.
Yeah, and one thing, this is like this whole thing is very, very, very complex, because like when I showed you that for those 4 stations, they didn't find an annual cycle for the Arctic. For their paper, it's true, but they measured at -20 and -25. So it's not true in general. And that shows how complex that is. You always have to look at where in the temperature spectra you are, what are the concentrations, and just something that's valid for one sort of INP at one temperature can be totally wrong at a different temperature. So it has so many dimensions, this whole problem.
Yeah. Are there any more questions? Or,
No, we don't.
Yeah, I think we should thank Heike for this excellent talk and interactions. I hope many students who may listen to this, the presentation is available, and actually they will take up some this important topic. It is quite important for our weather prediction and climate, and so let us hope and
yeah. And what I will do, what I will do now is that I will take those publications you sent me, I will put them in my literature list. I will also include that Gary Mella paper and that Worthy paper with ammonium sulfate.
Yeah.
I hope I don't forget anything. So just to be fair, I just put that all in. And yeah, and then you just pronounce how that.
Yeah, we will make it available through a link at the just beside your talk, where we are giving the link, so that everyone can access that and
okay.
do further.
Yeah. So thank you very, very much to like Mercy, Mahen, and to you, Tara, and for organizing that whole series and for everything, really. So
thank you.
It was very, very nice.
Thank you so much, and thank you for Anupam and Aran, who has participated also in asking several questions, and thank you so much, and thanks to Mahen.
Thank you, Dr. Heike. Thank you.
Okay. Thank you.